Okay, so welcome to the session on the life science and social science. My name is Professor Natachai Sisawat. I'm from Faculty of Medicine,ฬาลงกรณ์ University, and will moderate and also be the chairperson for this session. Today we have 7 research funding from Thai Asahi during the past year, and we would like to start this session with the first presenter, which is Dr. Panaya. The first presenter will using the video presentation. He's not here today. His name is Assistant Professor Dr. Drew Bennett Mallory from Sassin Graduate Institute of Business Administration. He will present the research on understanding the ecosystem of recruiting people with autism in Thailand. Professor Drew, please.
Hi, everyone. This is Dr. Drew Mallory of Sassin School of Management. I just wanted to welcome you to my short presentation on the research that I did with the support of the Asahi Glass Foundation. I'm very pleased to be able to present on the publication capable of much more, the effects of vocational training on caregiver expectations for neurodivergent dependents in Thailand, which was made possible, of course, with the Asahi Glass grant. I was willing to present this 2 years ago and reached out to the office several times, but it was very difficult to get in touch, and I'm sorry that the report which I sent was not presented to the Asahi Foundation. I still have that report if you're interested in it. The goal, of course, of this presentation is just to explore the overall findings of the research and to explain how vocational training really changed the perception of the limitations for Thai families and expatriate families on their neurodivergent children and their capacity. So, of course, I would like to begin with an acknowledgement and a formal apology again for not being able to present in person. I wanted to express my sincere regrets and just offer my formal apologies for being unable to present in person. Each year at the time of the Asahi Foundation's presentation period, we have our international conferences, and I am unfortunately unable to be present in Thailand for any reason during that time. So I feel very much sincere gratitude, however, for the gracious support in 2024 for this project, and the support of the foundation has made possible the in-depth exploration of Thai caregivers and the caregivers of expatriates living here in the country, as I hope that the presentation will make clear. So let's begin by looking at the stark reality of neurodivergent people in Thailand today as we jump in. So the neurodivergent landscape in Thailand is quite distinct and different from other places in the world. Globally, 15 to 20% of people are neurodivergent. And by neurodivergent, what I'm talking about is people with autism, with ADHD, and with a number of other conditions as well that you might be familiar with, like Down syndrome, etc. All these people would be considered to be neurodivergent, which is why we have a very high number, around 20%. Around 1 in 100 people have autism. Autism does not look any one particular way, but what we do know about people with autism is they're very unlikely to have employment, regardless of the severity of the condition itself. Now, here in Thailand, we have official estimates of around 500,000 to 700,000 cases in Thailand, but that is not what the official estimates say. They say around 4,000. So for some reason, the government is not very clear on actually how much the cases of autism are in Thailand in general. Only 15% of people who do have neurodivergence have access to basic health care in Thailand, and this is because of the way that it's required to get diagnosed. You have to get a disability card, and for many, many reasons, people are not willing to get that. And so you see people with autism not actually having the types of care that they need to get their basic needs met, and certainly not to be able to get things like training that they would need in order to successfully get healthcare in the future. And we can take a look at the difference between the reality and the official records. There are some very specific cultural reasons why that is the case. So we can see that the architecture of stigma in Thailand is very strong. This showed up in my own research from this study, but also from research that I do outside. And there are two forces in Thailand that really combine to create a kind of toxic atmosphere for many people who are neurodivergent or experience other types of differences. One of those is karma, a fatalistic belief about the nature of any difficulty or disability that you have. And the other is face or nata. So on the left side, we see karma here. In Thai is this spiritual belief. Caregivers typically see karma as a consequence of sin, either their sin or the sin of their child. And that is not just a spiritual belief. It really frames neurodivergence as a punishment, and it's a punishment that cannot be changed. On the other side, we see nata, this idea of face, this intense pressure to protect your public image and the public image of your children or your families. The result is that families will actually conceal the diagnosis to avoid shame, and that leads to critical delays in early intervention, but it also leads to other things that we see later on, which is preventing children from being able to achieve their full capacity. Now, this cultural stigma does not just stay at home, it also affects corporate policy as well. So we do see that there is definitely a policy gap. And there are some research questions that this led to in my case. The core idea here is that cultural stigma drives companies to pay billions of baht in fines rather than to actually hire neurodivergent talent. And why would a company hire a neurodivergent person in Thailand to begin with? Well, there is a legal mandate in Thailand to fill 1% of a workforce for your company if your company is over a certain size with people with disabilities. And one registered disability is autism. But because people believe in Thailand that neurodivergence is a curse because it's a consequence of sin, even though we have a 1% requirement, people will typically believe that a person with disabilities, and this is not just autism, but that they can't actually do meaningful work. And because of that, because they think their companies are not set up to handle someone with a disability, they will pay a fine so that they don't have to hire someone, right? They don't want to hire them, and so they don't. So the law says 1% of people with disabilities need to get hired. The reality is companies won't do it. The paradox is that families are avoiding getting those disability cards, which would allow them to then become part of that 1%, which is also a big problem. So our question, one of our questions was, how do Thai caregivers of transition age neurodivergent adults view employment preparedness in a very broad sense? The second question is, how are they using the existing services and how do those services impact their perception? So when parents do reach out or caregivers do reach out to a service like the one that we worked with to get care, does that affect their perceptions? And we did this in a very rigorous, culturally adapted survey. This started out with a hybrid inductive-deductive framework to balance EdTech, which is outsider knowledge, and EMIC, which is insider knowledge, meaning that I am an outsider, of course. I'm also not a person with autism, and I'm not a person who's caring for people with disabilities. And so I am an outsider. So we worked with people on the inside, meaning caregivers, but also meaning service providers, to make sure that we built a good and proper study that could reflect their lived experiences. This was validated by subject matter experts in Thailand as well to make sure that we were capturing things properly. And what we really wanted to do is make sure that we understood their story. So we started out with a literature review that helped us to create a protocol that we used to create focus groups and focus group questions. And that led us to then interview different people to try to get an understanding of what their experiences were like. Now, the focus groups included Thai and expatriates who had not yet received any care, and we wanted to understand what were their concerns and what were they looking for. The interviews were people who had already received care and their children had now gone out, graduated the program, and tried to get jobs, and we wanted to see what was happening. Throughout that process, like I said, I consulted with subject matter experts to make sure that we were on track, both in terms of the protocols, but also in terms of the findings and the results. As I mentioned, we had two different cohorts and two different environments. Both of these were, of course, approved by the IRB. The cohort A was the pre-training group, so 10 caregivers came in and spoke with us in a very sensitive environment, but a very important one, to talk about what it was like to have a child with a disability, particularly autism or a similar condition. And then 13 one-on-one interviews were conducted later in our studio at Sasen to talk about their experience. All those people in the second group had completed a 2-year vocational training program. And we considered this to be action research because we wanted to be able to record that content so that we could use it later to create training videos if people wanted to. Not everyone did, but people did ask for that. They wanted to make an impact. And so we gave that to them as well. And the funding from Asahi helped to cover that. In terms of the demographics, I'll just quickly go through this, but we can see that the majority were, as you expected, around 50 or so years old or older. They had children, of course, and they've been taking care of them for their whole lives. We had about 50% were expatriate Thais, and Thais rather, and the rest were expatriates, and some of them were mixed as well. And the majority of people had people with autism that they cared for, so autistic people. In terms of looking at their findings, I've highlighted a few of the examples for you here just to show you what some of those findings look like from the early stage of coding. We wanted to go through and code the content in various ways to try to make sense of it. And these are some of the themes that came up. One was making sense of fate. So people spent a lot of time trying to explain karma as being a punishment and eventually moving from seeing it as a punishment to seeing it as an opportunity to create merit. In the beginning, karma was a sad thing. Later, they said, okay, well, maybe karma can actually promote tolerance in myself and promote tolerance in other people. So people definitely saw in both groups karma as something that was important, but their views changed over time. Secondly, we saw a lot of medical gatekeeping that they reported on the outside, where doctors dismissed the potential of their children, treating it as a physical limitation, where they did not ever get opportunities because the doctors prevented that. And thirdly, we see a family paradox where extended family also tried to hide the issue. They didn't want to talk about it. So they said everyone in the family was, in this case, sorry, the quote was kind of cut off, but was suffering essentially at this time. So people loved the children, but they didn't want to give the support. And when we compare that to the post-training group, we do see a very different transformation. This is the caregivers from the first group. In the second group, we see that things start to change. So in the pre-training group, people had a lot of doubts. They had a lot of concerns, a lot of worries. They didn't feel like their children were going to be capable of making a lot of progress. They were worried about isolation. They were worried that their children would be dependent on them forever. They were very helpless. They were searching for answers. It's very difficult to get information in Thailand about anything, but especially if you have a need like this. And in the post-training group, we see a huge transformation. People are communicating. Children are communicating. Children are independent. And the view of the children, which is the most important thing, had changed. The parents no longer saw the children as being incapable. They no longer saw the children as being not able to do anything. Something really changed in the training process. And one specific caregiver actually sums this up very well. I'll let you see. This caregiver said, I realized she was capable of much more than I had thought, and that's the name of the paper. So caregivers ended up explaining why their views changed as actually being the training program's individualized support and opportunities. But it wasn't just the training itself that made the difference. It was watching their children actually see that they had abilities. So the children recognized that they had abilities, and eventually the parents saw that as well, that the children actually had abilities, and that led to something very unique. And not everyone got jobs, but the parents universally saw their children as being very different. Other things that happened as well, I would say, as you can see here on this chart, in the beginning, that parents started out with a very medical mindset, looking at autism as something that was problematic. It was something bad happening inside the child. Later, you see on the other side, parents saw society as the problem. That actually what was happening was society, including themselves, the parents themselves, were preventing their children from recognizing their full potential. And that's really what ended up happening. The training allowed people to overcome the social barriers and let the ability that they had internally out. And that was very unique. So the ripple effect that comes from this is that first individuals build their own personal efficacy, and this then affects caregivers in eventually being able to see their children as having abilities, and then eventually at the very end, the parents became very involved in social advocacy as well, pushing for change at the government level, and some of them actually did achieve nation-level change, which is incredible to be able to capture that as well. And the findings are, of course, powerful, but we do have to view that through a very transparent lens. This study was limited, obviously. It was the first of its kind in Thailand, so it was very limited. It did have a very Western-centric lens because of me and because the training that they received was very Western-centric. The strength is that it's quite unique. Nothing like this had ever been done, like I said, and it has very practical implications for caregiver trainings and for policy here in Thailand, and we did share this with various bodies here in Thailand as well. The conclusion here, of course, is very practical and very significant. We proved that neurodivergent individuals in Thailand are capable of more. We documented that. They proved it themselves. We were able to do this, of course, with the support of the Asahi Glass Foundation, and we're very appreciative to that, to be able to say thank you for supporting this profound work. Thank you for supporting the families of people with disabilities and for supporting the people themselves.
Thank you very much, Dr. Drew, for a nice presentation. And because he's not here, so we'll skip the Q&A session and move to the second presenter. The second presenter will be Dr. Panaya Kochaplai from the Institute of Biotechnology and Genetic Engineering. Will present on the topic bacterial polymer for soil restoration, the effect of extreme weather event on physical chemical properties and microbial community of soil amended with biopolymer producing bacteria.
So thank you very much and good afternoon everyone. So if we talk about environmental issues, so what's come to your mind? Maybe now PM 2.5, maybe plastic accumulation in the ocean. Those are all vital issues. However, like there is another silent crisis that people are less aware of, and that is land degradation. Okay, so land is a finite resource. So we are already dealing with problem soil, which are like naturally unsuitable for agricultural activity. However, together with our human-driven degradation, so we now reach to the point that over 1 billion people are experiencing lower crop yield due to land degradation. So if we talk about the properties of problem soil, so it varies. It can be like low fertility, soil compaction to low water retention. So if we want to improve soil nutrients, so we add fertilizer. But if we want to improve soil water retention, one approach is to use superabsorbent polymers or SAP. So these are polymers that can absorb and slowly release water available for plants. However, SAPs we use now is chemical acrylic acid-based synthetic. So there is growing concern that as they degrade, the accumulation of the residue might pose a long-term environmental risk. So in our lab, we think about like what about like less toxic polymer with similar property and can be produced biologically. So many of you are familiar with natto, the traditional Japanese soybean product, right? So if we talk about natto, some of you may think about its strong smell, and some of you may think about this slimy string. So the main component in this slimy string is the polymer called polygamma glutamic acid or gamma PJ. So you can, So you can guess from its name. The gamma PJ is the polymer of amino acid glutamic acid. It can be produced by several strains of bacillus. So the property of this polymer is that gamma PJ is biodegradable, biocompatible, non-toxic, and also one interesting characteristic is the gamma PJ is hygroscopic, so it means it can absorb moisture from the surrounding environment. So according to this property, gamma PJ has been widely used in various applications, including agricultural area as a fertilizer synergies and also the precursor for sap production. So many studies have demonstrated that the application of gamma PJ can enhance soil moisture retention, can promote stress tolerance in plants, and also can support plant growth. However, the challenge of using gamma PGA is first thing is the high production cost due to the reliance of the substrate, which is glutamic acid, the precursor of the amino production, and also like the downstream process. And gamma PGA can be degraded rapidly, so we need reapplication of gamma PGA of this polymer into soil. So our lab solved part of that bottleneck. So we isolate Bacillus subtilis FSO3 from Tuanao, which is Thai fermented soybean from the northern part of Thailand. So this strain is glutamate independent gamma PGA producer. So it can produce a certain level of gamma PGA without the need for external glutamate supplementation. So it can synthesize glutamate, the precursor for gamma PGA, directly from the carbon source. And this strain is quite tough, so it can survive and also continually produce gamma PGA under several stress conditions. Okay, so our previous work demonstrated that its fermentation medium can prolong soil moisture retention, and it can support, it can enhance seed germination. And now we are developing what we call soil amendment. So we try to combine this strain together with agricultural residue like palm empty fruit bunch, which can act as a carrier for bacteria and also the substrate for in situ gamma PGA production in soil. So it seems like we have a promising soil amendment. So one more issue come to our mind. So what about its efficiency under the extreme weather event? So climate change is bringing more frequent droughts and flood. So not at a separate event, but sometimes as an alternating event on the same land, and sometimes even in the same growing season. So actually, like the soil we use, we collect it from Nan province, the northern part of Thailand. And 2026, if you can remember, Nan 13 province in the northern part of Thailand experienced severe flooding. And right before this flooding, Nan experienced what we call it prolonged drought in the dry season. So you can see in the same area, it can face with many extreme weather events. So our quick key question is that, so what about our amended soil? So can this microbial-based soil amendment maintain its efficiency under drought and flooding? Okay, so our objective is quite straightforward. So we want to evaluate the effect of our soil amendment on soil physical-chemical property and microbial community under drought and flooding. So simply we prepare two types of soil amendment. A is palm empty fruit bunch together with yeast extract as the nitrogen source, and B is the same mixture inoculated with our strain Bacillus subtilis FSO3. So after 14 days of incubation to allow bacteria to produce gamma PGA. So we mix this soil amendment with soil and subjected to them to 3 water treatment. So normal watering, so we water it every other day for 10 days. Under drought conditions, so we withhold irrigation, and under flooding, so we submerge this soil pot under standing water. After that, we collect soil sample and submit it for physical-chemical property analysis and also amplicon sequencing to investigate change in microbial community. I'm sorry for the clouded slide. So for soil physical-chemical property, if we take a look at from what we call, okay, the addition of palm empty fruit bunch could enhance like around 3 to 4 fold of soil organic matter across all water treatment. And when we take a look at water retention, so if you focus here, FC is field capacity. It is the maximum amount of water that soil can hold. So the addition of palm empty fruit bunch alone could enhance around 12% of field capacity. However, co-amended with our strain can further increase field capacity, especially under drought and flooding condition. And if we take a look at what we call plant available water, or PAW, which is the difference between field capacity and permanent building point, we found that co-amendment with Bacillus subtilis FSO3 could maintain higher PAW under drought and flooding conditions. So it may suggest that the benefit of using this strain is more pronounced under the water stress condition rather than under optimal condition. Okay, so we found that in unaminated soil, it had the highest bacterial diversity. So let's take a look at changes in microbial community. So for unaminated soil, we found the highest bacterial diversity. However, this diversity dropped substantially after the addition of palm empty fruit bunch. And under drought conditions, so we found like drought decreased bacterial richness across all treatment, whether it bacterial community or fungal community. And if we take a look at under flooding condition could enhance what we call fungal richness and also its phylogenetic diversity in fungal community. Okay, and we found that the sample from the normal and drought sample cluster together. However, flooding samples separate from both cluster and from each other. So let's take a look at the changes in bacterial community under normal water and drought. So the dominant bacteria in unaminated soil is the slow-growing oligotope, which was nearly absent after the addition of palm empty bunch. And interestingly, under the drought condition, we found that in the AD treatment, so without our bacteria, the community replaced entirely by the spore-forming firmicutes. However, in the BD treatment, so we add our strain. The dominant bacteria are providencial, which is a metabolically active proteobacteria, whereas the spore former largely absent. So we think that it seems like the gamma PJ can maintain sufficient soil moisture to sustain a metabolically active probiotrophic community rather than driving this community to dormancy. Okay, and under flooding conditions, so flooding conditions shift both bacterial treatment under AF and AB toward low oxygen adapted bacterial taxa, and that in the BF treatment, so the dominant bacteria are the fermentative, obligate fermentative and aerobe. So this explains which can produce organic acid. So this explains the lower pH in the BF compared to the AF. Okay. So the key findings from this study are that we found that only PFB amendment is enough to improve soil fertility and water retention across all moisture conditions. However, co-amendment with Bacillus subtilis FSO3 can further enhance this field capacity and also plant available water, especially under the drought condition. Okay, and under drought condition, so we found that gamma PJ itself can play an important role in maintaining metabolically active bacterial community rather than driving all the bacterial community to sporulation. So, and under flooding conditions, so all bacterial community shift toward the anaerobic bacteria. So since my time is up, so thank you very much, everyone. And okay, so I would like to thank the 2025 Asahi research grant from Asahi Foundation and also like Assistant Professor Tim Sewitiyayon and Assistant Professor Wan Wisatansak from Mahidol University for their comment and suggestion about this soil experiment. So thank you.
Thank you very much, Dr. Panaya. Any question from the floor? May I ask you one question? Because as part of in medical field, we have many infectious disease that transmitted by people exposure to the soil and they have some wound on the leg, such as one of the important disease in the southern part and northeast of Thailand, like leptospirosis or โรคฉี่หนู and also melioidosis that directly transmitted from the soil contaminated with the bacteria Pseudomonas. Do you think this kind of the technique can help us also to prevent or mitigate the transmission rate of this important bacteria or any literature talking about this thing?
So I think it depends on the strain that cause the infection. So, but one thing in our lab we are doing the functional characterization of like the prediction of this group of bacteria. So is it like animal pathogen or plant pathogen or not? But if we to conclude that it can prevent or enhance the occurrence of the emergence, it's still, I think it's still inconclusive because we are in very early stage. But actually, I think like since under flooding condition and the addition of gamma PJ can enrich very diverse, what we call like fungal family. So initially, that is one of our concern. So even though it can improve soil property, but if it enrich pathogenic fungal community, so it might not be like. So that's what we keep in mind.
I think maybe we can, if you can explore this one, should be a lot of impact to medical field because other than be giving like antibiotics to the people, but we just modify the environment to be the unhealthy environment for the bacteria. That might be another approach for the future. Thank you very much.
Thank you.
So we will move to the third presenter. It will be Assistant Professor Dr. Granok from Faculty of Medicine,ฬาลงกรณ์ University, Department of Parasitology. Dr. Granok will present the using a One Health metagenomic-based approach for surveillance of emerging zoonotic pathogen in blood-sucking insects in tourist cave in the northern part of Thailand. Dr. Granok, please.
Okay, so can you hear me?
Yes.
Let me test. Okay, thank you so much. Hi everyone. Thank you so much for giving me an opportunity to give a presentation about my work. So I'm a medical parasitologist who work on the vector biologies and vector-borne disease. So I have a special interest on the emerging zoonotic disease in Thailand, especially leishmaniasis. And I would like to apply a new technology about the sequencing technology to explore the pathogens in the blood-sucking insects in the tourist cave, especially in the northern Thailand. So I apply the One Health concept to investigate the relationship between the animal, human, and environment for disease transmission. And I select the northern Thailand because that the northern Thailand have a very nice place for tourists, right? And for the tourism as well, and have the ecology that suitable for disease transmission. And I select the cave ecosystem for this work. So I have a special emphasis on the sandfly biology as well, because that sandfly is a minute blood-sucking insect. It's different from mosquito. So it can transmit a lot of disease. In this case, is leishmaniasis. And leishmaniasis is an emerging disease in Thailand that's actually leishmaniasis is distributed worldwide. But recently in Thailand have been reported by the WHO as the endemic area of leishmaniasis. And this disease transmitted by sandfly, as shown you in the previous slide. And the pathogens will transmit to the people through the bite of female sandfly and cause a disease in the three main forms. The first form is a cutaneous leishmaniasis, have the ulcer, is a skin ulcer, and the second presentation is mucocutaneous leishmaniasis, have the destruction of the mucocutaneous tissue, especially the facial area. And the most severe form is visceral leishmaniasis that can cause a prolonged fever, severe pancytopenia, hepatosplenomegaly, and finally the people is more likely to get death if untreated. So this disease is important emerging disease in Thailand, and we support that it might be transmitted through the ecosystem that have sandfly and animal and human interface. So just would like to introduce you about the endemic area. In the northern Thailand is the endemic area of leishmaniasis. Not only in the northern Thailand, in the southern Thailand as well. So I select the Chiang Rai province because that is the most endemic focus of leishmaniasis in northern Thailand. And okay, just show you a picture of the case have the cutaneous lesions and have a mucocutaneous lesion as well. And this case, this patient reside in the Chiang Rai province. And then I collect the sandfly from the cave, two tourist cave in Chiang Rai, and we collect sandfly from the two cave and one livestock farm. So I showed you that we place the CDC light trap in the cave and in the livestock farm as well. And after we collect the sandfly, we identify the species by their morphologies, and we know that the over X, around X species identify, and most of them it belong to the Sergentomyia species that feed on reptile. But some of them is Phlebotomys species that feed on human and mammal. That means in the cave have the human blood-sucking insect, and this can increase the risk of the disease transmission. Okay, so this is an experimental workflow. After we collect the sandfly, we extract the DNA from the sandfly, and we identify the species through the morphology of the sandfly teeth and spermatica. And after that, we apply the high throughput sequencing to see the presence of the pathogen as well as the bacterial endosymbiont in the sandfly. Okay, so just show you the morphology of the sibarium and spermatica of the sandfly that we collect from the cave. You can see the different morphology of each species. This can enable the morphological species identifications, and we found that Phlebotomys stantonai is the most abundant in Patong cave. That is a tourist cave, very famous in Chiang Rai. And Phlebotomys stantonai have been known to feed on the mammal and bat also, because that in the cave have a lot of bat. When we collect the sandfly, we observe that a lot of bat reside in the cave. And after investigation, we found that a lot of sample of approximately 300 sample, around 70x sample test positive with the Leishmania DNA, accounting for the 26% have the Leishmania DNA is potentially have a natural infection of Leishmania parasite in this sandfly. And not only the Leishmania species, we also detect the bacterial endosymbiont as well. Wolbachia cardinium, and we can also detect both bacterial endosymbiont in this sandfly. And we do the association analysis. We found that when sandfly have the Wolbachia infections, is likely to have no Leishmania infection. That means the Wolbachia can influence the Leishmania establishment in the sandfly, and this can can have an implication for interrupting the leishmaniasis transmission in the future. So after we do association analysis, we found that in the case of Wolbachia only infection have a low odds ratio and have the significant p-value as well. That suggests that Wolbachia might indirectly influence parasite persistence in sandfly and potentially through the upregulation of antimicrobial peptide and host immune activations. Okay, we also investigate the genetic diversity of this bacterial endosymbiont through the haplotype analysis network, and we found that they have the substantial genetic divergence of Wolbachia in sandfly, and we found the 3 clusters in this network, and this show The evolutionary relationship of Wolbachia is linked to the Drosophila associated lineage in Q-leg pipian mosquito as well, and the parasitoid wasp is suggesting the horizontal transmission of Wolbachia in the sandfly can be obtained from the ultra-dipteran insects in the cave. And we also explore the structural analysis also, and we found that in the some haplotype of Wolbachia endosymbiont have the insertion loop in the Wolbachia surface protein, and this protein responsible for the host interaction. So that means sequence variation in Wolbachia surface protein can influence the host interaction and contributing to the parasite persistence. And we found that in this sequence have the four hypervariable regions in this, and we found the spatial insertion loop in this. This is the first discovery on the Wolbachia surface protein in Thailand. And we also explored another endosymbiont as well, not only Wolbachia, we found the cardinium infection in sandfly also. And thank you so much for great contribution from Asahi, because that I submit my paper into the highly reviewed journal Parasite Vector last night, and I acknowledge there's a partial support by the Asahi Glass Foundation. Okay, thank you for our collaborator as well for our research. Thank you so much. Do you have any question?
Thank you very much, Professor Dr. Kanok. Any question? May I ask you one question? Because Wolbachia is also not only interested in the field of leishmaniasis, but also in the field of dengue infection. And people use the Wolbachia as one of the tools to reduce the transmission of dengue virus in the mosquito. So how we can do that? You also mentioned in your presentation that the Wolbachia may be one of another tools to reduce the transmission of the leishmaniasis. This kind of thing can implement into more in the kind of the translational research or implement into epidemiology field.
Exactly, because that I think that this is a new discovery of Wolbachia in the oval sandfly. But from the previous literature, it needs the further investigation, because that a lot of papers have the conflicting result. Because there some papers show that they show co-detection of Wolbachia with leishmania, but in the different sandfly different sandfly species in the different endemic area. But for in Thailand, we found there's a negative associations when have the Wolbachia infection is likely to have no leishmania. So that means so we need to investigate more by the establishing, maintaining sandfly in the laboratory and try to infect with Wolbachia and leishmania together to see the relationship between endosymbiont and parasite.
Thank you very much, Kap. So we move to the fourth presenter. The fourth presenter will be Dr. Jirasin Kuntongkeo from Department of Microbiology, Faculty of Science. Dr. Jirasin will present the research on developing coffee berry beverage production from spent coffee pulps using flavor-enhancing strain of Saccharomyces cerevisiae. Dr. Jirasin, please.
Good afternoon, everyone. Let me start my presentation like this. So, how many of you guys in here like to drink coffee? Okay, I need to put me as well. You know, it's an adult beverage that help us survive in the hard working day, right? But how many of you know how coffee is produced from? So here is the coffee cherries. Coffee is actually the berries plants. And here's the coffee cherries. And the coffee that we drink made from beans here. Here's the beans of the coffee and the center of the coffee berries. So to process these coffee beverages, they need to pulping out. Here's like the pericarps. structure, the outer skin, the pulp need to pulp out, and these are the pulp that normally they throw away as a waste. So in coffee processing, they're gonna get a lot of these waste, the coffee pulp or coffee husk. And these coffee pulp or coffee husk contains a lot of tannins and carbohydrates. They rich in the nutrients, so very equal toxicity. And normally in Thailand, the producer, especially the small producer, they normally throw away these coffee pulp. And yes, it's gonna accumulate a lot of pathogens, gonna get unpleasant odor, and also leads to a lot of human health problems as well. So when we consider this coffee pulp chemical composition, the previous study found that these coffee pulp pericarbs contains a lot of fermentable sugar, especially the sucrose, glucose, and some of fructose. And also they contain moderate contents of proteins and lipids, together with some tannins, anthocyanins, which are more similar to the wine, so the fruit wine, the grapes. So from the chemical composition, this coffee pulp meets the criteria for fruit wine production. So the key idea of my study is we're gonna use these coffee pulp that normally throw away and ferment them into coffee pulp wine using the yeast. So talking about fermentation of wine, very important microbes that prey in lows. We call it the yeast. Technically, the name of this yeast is Saccharomyces cerevisiae. And as you can see in this picture, this is the commercial or typical use in wine fermentation industries. The Saccharomyces cerevisiae EC1118. These strains or these yeast strains used in commercial wine production, these yeast can ferment, can convert the sugar into high concentration of ethanol and leading to the consistency of the products in industries. But the limitation of using these Saccharomyces yeast alone is that it's going to homogenize the flavor, which means normally Saccharomyces cerevisiae have the limit capability to produce the flavor compounds. So the product will be homogenized and will be get boring from customers. So to fix this, I would like to introduce another group of yeast to you. Its named non-Saccharomyces yeast. In biotechnological application, we normally classify yeast into two main groups. The first one is Saccharomyces cerevisiae, as I said, can produce a lot of ethanol. And another group, we call it non-Saccharomyces yeast. And yeah, from the name, it's not Saccharomyces cerevisiae, like Curomyces marcianus or Hansenia spora uvarum. You don't need to remember this name. We don't need to take exam for this, okay? But the interesting characteristic of these non-Saccharomyces yeast is that they can produce a lot of or produce diverse secondary metabolites which impact the flavor compounds like the high alcohol ester here, these compounds called ethyl isobutylate contribute to fruity flavor that can sensed by human. And another compounds group is called terpenes group, like this limonene. Limonene will contribute to lemon zest flavor compound. So the idea of my work, the solution of my work is that we're gonna use, as I said, we're gonna use the spent coffee pulp or coffee husk to produce wine, but to diversify the flavor compounds in wine, we're gonna use the co-fermentation technique between the Saccharomyces cerevisiae EC1118, the commercial for fruit wine, grapes wine fermentation, with the flavor enhancing non-Saccharomyces yeast. So let's get to the results. First, I need to screen or isolate the non-Saccharomyces, the flavor enhanced non-Saccharomyces yeast first, though technically not me, my graduate student. So we can isolate, and here we can isolate some amount of non-Saccharomyces yeast isolate. For example, 313, 315, 16, 17, and 19. And we measure the fermentation ability. As I said, Saccharomyces cerevisiae dominate the fermentation. So we like to get the non-Saccharomyces yeast which have a comparable fermentation ability with Saccharomyces cerevisiae. And the result here, the control is here, EC1118, this graph, this blue graph, and I got one green graph that got the highest fermentation ability and technically higher than the Saccharomyces cerevisiae as a control. This isolate named 319, JK319. So this one have highest fermentation ability. Then when we check the phylogenetic analysis using ITS region analysis, we found that isolate JK319 belongs to Cucurbitacillus marcianus yeast with 99.9% similarity with the reference strains. So what is Cucurbitacillus marcianus? It's not Saccharomyces, but this K. marcianus also holds the generally regarded as safe or glass status for consumption. It's also widely used in biotechnological application as well, as similar to Saccharomyces cerevisiae. So technically using this strain for beverage is safe for customers. And also we analyze the sensory analysis using the descriptive, which means we smell the fermented broth by certified sensory panelists, and the panelists describe the sensory like get some citrus and lemon peel note, which is very interesting characteristic for producing beverages. So next we produce the coffee pulp wine. Yes, we use coffee pulp, adjust the bleach total sugar, and then ferment them. So I would like to go into this. The fermentation would divide it into single strain fermentation using Saccharomyces cerevisiae as a control, using Cucurbitacillus marcianus single strain. And the next are the co-fermentation. We co-ferment them using two different techniques. The first one called simultaneous inoculation, which means we inoculate Saccharomyces with Cucurbitacillus together. And the next one is the sequential inoculation, which means we inoculate Cucurbitacillus first, let it grow for 24 hours, and then inoculate Saccharomyces. And here's the result. Sorry for the slide. Technically, the appearance of all coffee pulp wine looks the same, like pale amber to yellow appearance. And then we analyze the chemical compounds. We found that the co-fermentation, whether the sequential inoculation or simultaneous inoculation, got almost similar ethanol concentration to Saccharomyces alone. This is a control. So the ethanol concentration is almost similar. And then we also measure the methanol content. You know, methanol is toxic to us. And the International Organization of Wine limit the methanol concentration for red wine not more than 400 milligrams per liter. And all of the wine here contains under limit. So which means methanol are safe. And importantly, we measure the volatile flavor compounds. We got a lot of flavor compounds from GCMS analysis. And I want to summarize like this. The simultaneous fermentation, we found that it could maximize the flavor compounds from using Saccharomyces alone. For example, here, isoamyl alcohol, these compounds contribute to banana-like flavor. And using Saccharomyces alone got around 332, but using simultaneous fermentation got like about 2-4 higher, and also a lot of chemical compounds were higher in co-fermentation. But the main problem is that I said it's higher, but whether we want to know that human can sense or not, is it enough concentration for human to sense? So we measure by using this value called odor activity value, or OAV. This one calculated by the concentration of each flavor compound divided by threshold value that human can sense. If the flavor compounds more than one, which means it contributes to aroma, and here the simultaneous fermentation, we got a lot of them more than one. But some flavor compounds disappear when we use co-fermentation. Is demonstrate that it suggests that Cucurbitacillus population was not enough. So to conclude, yeah, we successfully produced coffee pulp beverages, and then we isolate the Cucurbitacillus marcianus, co-ferment them, and simultaneous co-fermentation was the most effective approach. So for further study, we might need to optimize the ratio of curomyces to Saccharomyces, like increasing curomyces marginous ratio, and also optimize the fermentation condition, times, temperature, and not to overinterpret this just laboratory scale experiment to sell out like a product's got a lot of things to do, like safety profile, FDA analysis, something like that. And I would like to thanks to Asahi Glass Foundation for financial support, and my colleagues, collaborators, Associate Professor Dr. Intawut, Professor Dr. Apani, Dr. Bumin, and also my students, especially Ms. Tamonwan, she's here, and Ms. Bobusha here. They all graduated, which are contribute to this project. So thank you so much for your attention. I appreciate to take questions.
Thank you very much, Dr. Jirasin. Any question? May I ask you about, you use the OAV as the outcome to indicate but do you have any data in terms of using human sensory perception?
Yeah, we didn't perform that yet. So as I said, it's just very first step of the study. So when we, after optimize the fermentation condition, fermentation inoculation, we're going to use the human to sensory these things and doing the heat map sensory using sensory panels. Yeah, it's going to be further studied.
Thank you. And in terms of the safety profile, you mentioned about the methanol things, but any other toxic substance that such as the tannin or other toxic agent that we should mention also?
Yeah, well, actually, for the toxic compounds, still have a lot of things like the fuel alcohol is like a higher alcohol, higher than ethanol. It's an alcohol that have more carbon atom than ethanol. If these compounds are too much, it's going to get toxic to customers. So we may need to take care of these things as well in further studies. Thank you so much.
Thank you very much. So we move to the five presenter. Will be Assistant Professor Dr. Sutlop Ratana Keurkangwan. Dr. Sutlop will present the development of data bank for cocoa supply chain in Thailand.
Good afternoon, everyone. My name is Sutlop Ratana Keurkangwan. Today I'm going to present to you in the topic of the development of the data bank for cacao supply chain in Thailand. So this project is in the duration of 1 year, gratefully funded by the Asahi Glass Foundation. So in this project, we have been collaborate with mainly under the collaborations of the Faculty of Integrated Agriculture and the Department of Computer Engineering, Faculty of Engineering. So to briefly sum up for the whole project is we're trying to convert from fragmented paper records to the living data bank. So the problem starts with the Thailand cacao industry has been expanded rapidly. However, the upstream data collection remains in the fragmented and paper-based. So it limits the aggregation, georeferencing, traceability, and analysis. So our work, this one, we are trying to design, build, and field evaluate a centralized data bank for the Thai cacao supply chain. So the development follow a user-centered design method over the 5 iterative phases, which includes the 2 field deployments in Chiang Mai and Prachuap Khiri Khan province. So the main problem, as I mentioned a little bit before that, most of the cacao farmers in Thailand, they do not record their own production data. So this also includes to the processor and collectors hubs that they keep their critical records just only in like small papers or in just the ad hoc spreadsheets. So their records are in the fragmented, lossy, and so it eventually becoming the effectively invisible for ourselves, like a researcher or policy maker. And also there are information gaps between the supply and demand. It's not equals, and the farmers they don't know the cost modeling for their own operations. And also, as a researcher, we don't get adequate the feedbacks, and also the farmers themselves cannot use their own records in order to decide when and how much they have to put the fertilizer, the pest control, or the harvest timing. Okay, so why we have to focus on the mobile data? Because of the study has been many studies already concluded that the mobile phone has raised the farm efficiency and productivity. And also in the horticulture, for example, in Boro rice, the phone bridges information gaps and speed the technology adoption. But on the other hand, there still be 4 adoption barriers that still block the adoption of the mobile phone in the agricultural user, mainly on the farmers. The first one is digital literacy for the farmers, because the farmers across the global, not only in Thailand, they are mainly they are in the aging society, especially in Thailand, or they are above the age of 55. And also about the connectivity that caused the frustration and abandonment, and also the cost benefit doubt for the farmers, and also the path dependence, which leads to the 3 objectives of this study. We are trying to build the centralized data bank for the Thai cacao supply chain, and also they design the mobile apps that let the farmers and processor access the data relevant to the cacao production and the supply chain. And also we're trying to do the on-site training for the local farmers in order to encourage and promote them the adoption in the remote areas. So for those in here who may not familiar with the supply chain of the cacao upstream supply chain, we have to start with the seedling propagation for the cacao plants and then do the planting. And we have to wait on the average about 3 to 5 years until the fully maturization that start the production. So after the production, the farmers harvest the cacao pod or cacao fruits, and then they peel it to use the white matter inside through the process of fermentation and drying. These 2 processes, we call it the process of the pre-processing. And after finish with the dry beans, it has sent to the processing, which will be processed further and eventually becoming the chocolate or cacao that we consume every day. So on the stakeholder matrix here in this study, we included 4 main stakeholders, including the farmers, the hub collector, the processor, and ourselves like a researcher or analyst. So along the supply chain, starting from the farmers, the inputs of their data, they usually input the data of the land size, the tree counts, or the fertilizer, how much they put. And for the hub and collector, of course, they collecting the data about the raw material that they get from the farmers, and the processor also span during the drying and processing period, that after finishing the product, like a dry bean, the quality, the grade, size. But for the researcher and analysts, we span all through the supply chain, starting from planting to the processing. So we have done the gap analysis for the analog status quo for the current situation before our project, that intervention. There are 4 main dimensions about the gap analysis, starting with the data management. As I mentioned, that the data that farmers store is fragmented. And for the coordination, the farmers mainly using the manual reporting, and especially the most importantly is the irregular updates. Like they update the data by their own views. Sometimes they don't update it. And also the accessibility, because a farmer cannot access to their own productivity history. Because of themselves, they don't have infrastructure for them in order to store the data. And capacity also, because of the limitation on the low digital literacy. That's why we are trying to propose a digital infrastructure in order to help these gaps. So this is our design system architecture. So here we are proposing mainly 2 sites, a mobile application for the farmer and a web researcher for the researcher, and connected in the central database. So it includes in the 8 data schema by connecting the physical spaces into the data schema. So we are under the implementation of 5 main phases, which is under the 2 field deployment, as I mentioned, in Chiang Mai and Prachuap Khiri Khan province. So the first 2 phases that we implemented is that there are 3 main participant types and 3 persona, depending on the literacy levels. And then we take them feedbacks and then we try to fix it. So the main contribution for the first revision is that we changed the language into a Thai localization language. We improved the readability, the simplified registration, and also the most importantly, we use the Buddhist era, the calendar, not the Christ era, as an application around the globe. This is a phase 3 that we put our revision version back to the site again in another place in Prachuap Khiri Khan, and then we see another friction occur, but we try to fix it. So the main contribution and the breakthrough for the second revision is that we change how the application thinks. Like normally the farmer doesn't think on the left side, which is the action first, but the farmer think in their own farms, and then inside the farm there are some plots. And then in the plot, they choose action. So we shift the whole application accordingly to the feedbacks. So this is an example of the application that we made. Some of the some example, the distinct example that we made the contribution. For example, the farmer, as they have some trouble to input the data in numerical data, so we propose the stepper button like +1, -1, +0, -0 for 3 texts. And also the land size, we let them put the numerical data or draw the map, and the application will calculate the land size for them automatically. So these are the system validation test results, because we have a quite limited time. So unfortunately, I don't have the time to explain this a lot. So the implication, to sum up, is that the application, we can be sum up that the paper can be replaced to the application, and the friction is occurred not by the technology, but because we don't understand the user quite well. That's why. And to conclude, this model, we can be the centralized data bank can be reproducible in other types of crop, not only in the cacao, and also it promotes traceability for the traceability food system, and also it's under the user-centered design and training for the trainers. For the future work, after the study, we found that the majority of Thai farmers already been familiarized with the line application. So we are trying in the next quarters, we are trying to promote the usage of a line OA instead of the use in couple together with the usage of the application in order for them to input the data more effectively. And also we are trying to promote we call it agricultural calendar, like what is right now the time of the year that they have to do something, make them like a recommendation, a push notification for them. So that will be it for my presentation. Again, thank you so much for, especially for the Asahi Glass Foundation for funding this project. So any questions or comment would be greatly grateful. Thank you very much.
Thank you very much, Dr. Sutlop. Any question from the floor? May I ask you regarding on the data governance after I think this project still running, but after the project's running for a while, how do you perform the data governance? How to keep this data, because it's quite a useful data set.
Thank you very much for your question, Professor. As this is a very good point for the data. Because of we gratefully get the funding from Asahi. At the beginning, before sending the proposal, we have been planning prior that we need to set aside some budgets for the data management on terms of the data, and on terms of the on terms of the financial side. And on terms of the operation, we've been still continuing to work with the farmers. And each time that we go back to the site, we return the data back to the farmers and tell them what is inside based on our data. So To sum up your question is that we are still ongoing, working and trying continuously to improve the farmers.
Thank you very much, Kap. So we will move to the next presenter, Assistant Professor Dr. Kanika Kantasuk from Department of Biochemistry and Microbiology, Faculty of Pharmaceutical Science. Dr. Kanika will present by Zoom in the topic of designing an in vitro evaluation of the folate alpha receptor specific immunotoxin with albumin binding domain for the treatment of lung cancer. Dr. Kanika, please.
Okay, my voice is clear.
Yes, we can hear you clearly.
Okay, good afternoon, everyone. I'm Dr. Kanika Kantasuk from Faculty of Pharmaceutical Science,ฬาลงกรณ์ University. First of all, I would like to express my sincere gratitude to the Asahi Glass Foundation for generous financial support which make my projects possible. Today, I'm very excited to share my study on the designing of an in vitro evaluation of folate alpha receptor specific immunotoxins with albumin binding domain for treatment of the lung cancer. Okay, why I forgot the lung cancer? Because the lung cancer is the second most common cause of death from the cancer worldwide. There are many of the risk factors to occur the lung cancer. For example, the smoking, passive smoking, and exposure to the radiation, including the pollution. Many scientists support that the PM 2.5 pollution can cause the lung cancer, so the number of the lung cancer patient is increasing every day. Okay, among the lung cancer, non-small cell lung cancer or NSCLC is the major type of the lung cancer. The treatment of the NSCLC, including the surgery, radiation therapy, chemotherapy, or radiation surgery. The treatment have the side effect. For example, hair loss caused by the chemo can kill the normal cell as well, and the nausea, vomiting, or increase the chance of the infection. So the new development of the treatment is needs. Targeted therapy is the one of the treatment for the NSCLC. Immunotoxin is the one type of targeted therapy. And what is the immunotoxin? Immunotoxin composed of the antibody that's specific to the antigen that's overexpressed on the cancer cell. The antibody will link with the potent toxin that act as the effector molecule. The antibody will bring the toxin kill, especially the cancer cell that express the antigen that's specific to the antibody. Focus on the targeting molecule. The targeting molecule can be the intact antibody or the other fragment of antibody, for example, the FAB or the SCFV. Even the smallest size of the antibody fragment or human variable heavy chain or human VH. That's the smallest part of the antibody that's still binding to the antigen. Okay. The increasing of the immunotoxin can be used in the clinical use, especially in the solid tumor. And the successful of the immunotoxin depend on many factors. For example, the target molecule. You need to have the antigen that's overexpressed only in the cancer that you need to kill and no express or lower expression on the normal cells. You need the antibody that act as the targeting molecule that's specific to the antigen on the cancer cell and no cross-reaction to the normal cells. You need to have the high potent toxin to kill the cancer. And also because to kill the cancer or solid tumor, the antibody should be have or immunotoxin should be have the long half-life in the circulation to have the enough time to kill the tumor. And you need to sufficient yield for patient dosing. Okay. In my project, I focus on the alpha folate receptor. And why? Because the alpha folate receptor is overexpressed on the NSCLC and other tumor, for example, the ovarian or breast cancer. This figure shows the distribution of the alpha folate receptor. You can see in cancer cells, the overexpression of the alpha folate receptor is distributed on the luminal surface with intact directly to the blood circulation and intact directly to the drug in the blood circulation. Otherwise, in the normal cell, the alpha folate receptor expressed in very low level in the apical surface, so that make the normal cell safe for the drug injection in the blood circulation. Okay, nowadays we have some of the immunotoxin enter the clinical use. It linked with the toxin that's made by the bacterial toxin, for example, the diphtheria toxin and the Pseudomonas enterotoxin. In this project, I focus the toxin from the Pseudomonas enterotoxin in the truncated version and de-immunize some amino acid to diminish the T cell response or reduce the antigenicity of the bacterial toxin, but still have the protein killing activity. Okay, because of the immunotoxin and other biomolecules have the short half-life in the blood circulation, so many researchers can find a way to extend their half-life by adding the biomolecule with the albumin binding domain or ABD to extend the blood circulation half-life. And why ABD can extend the circulation but half-life? The first one, after fused with the ABD, the biomolecule will be large or big enough to avoid the get rid of via the kidney. That is the one reason. The second reason is the biomolecule with the ABD will bind with human serum albumin and go through the FC neonatal translocation recycling molecule in cell and to blood circulation and recycling again in cell, go to in cell and go to the blood circulation like this. So resulting the extend the half-life of the biomolecule, including the immunotoxin. Recently, I and my group developed a human antibody fragment that's specific to the alpha folate receptor. And we found that this human variable heavy chain domain, the smaller size of the antibody part, still have to retain the antigen binding affinity. Okay, because the smaller size, okay, this is the VH. This figure shows the VS. Where is the VS? This VS fragment have the smaller size and high tumor penetrated and can produce easily and high yield in the bacteria expression system. So we decide to go to this antibody fragment, the VH antibody fragment. By select from the face display library, we can select the 5 or 4 to 5 candidates of the antibodies specific to the folate alpha receptor and the clone 3A12102, or we call briefly VH, show the high potent binding ability to the folate alpha receptor antigen. So we go with this clone or this VH clone. The VH show they still bind to not only recombinant folate alpha receptor, but they can bind the folate alpha receptor that's expressed on the surface of the lung cancer cell line. And also they can bind to the antigen folate alpha receptor on the NSCLC lung cancer patient-derived primary cancer cells. This confocal analysis show that our VH antibody bind to the surface of the lung cancer cell lines. So at this point, we know that the VH show the potential to be the targeting molecule. Okay, and under supported by the Asahi Glass Foundation, our project is able to be proceed on the design and evaluate the in vitro ability of the folate alpha receptor specific immunotoxin with albumin binding domain for treatment of the lung cancer. Okay, we further evaluate the internalized characteristic of the VH because to bring the drug or toxin to kill the cancer cell, the drug could be internalized into the cancer cell. So we determine the internalization activity of our antibody, and we found that our VH antibody fragment can internalize into the cancer cell that's overexpress the folate alpha receptor. So we go with this antibody to be the targeting molecule in immunotoxin. We designed two versions of the immunotoxin. Our antibody ABD, albumin binding domain, to increase the half-life of our immunotoxin, and the toxin link with the first one link with the flexible linker glycine serine. We call the GS immunotoxin. The second format is linked by the EA linker with its rigid amino acid linker. We refer EAIT. We compare two types of the linker in the term of the production yield and the ability to separate these three domains to get separate these three domains and evaluate the binding ability to alpha folate receptor and albumin binding protein still have when we link with the difference of the linker. In terms of the expression yield, we found that the GS linker or immunotoxin link via the glycine serine flexible linker show the higher expression yield of the immunotoxin when compared with the EA version. So in this point, we know that the type of linker can affect the production yield. Okay. And after that, I determine the binding ability of the immunotoxin with the both of the alpha folate receptor recombinant protein and the alpha folate receptor that expressed on the cancer cell, and also determine the binding to the albumin. And we found that in the EA version show the higher binding ability to the all of the parameter, both by to the alpha folate receptor on the surface of the cancer cell that I show you,นะคะ. And also by to human serum albumin. EA version have the higher affinity when compared with the GA version. And so the next one, I go to the EA version to determine the cell cytotoxicity. Okay. The superior binding activity of the EA version may be contributed to the EA linker format. A rigid linker format can help to preserve individual function and can separate individual function, minimize the steric interference between antibody and ABD domain and toxin. So the 3D structure can imply that the steric rigid linker can totally separate three domain and not interfere with other. The last one, I determine the in vitro cytotoxicity or the ability to kill the cancer cell of the EA version among the various types of the cancer cell which express the alpha folate receptor. And we found that the IC50 value or the concentration of the immunotoxin that can kill half of the cell line is about 2 nanomolar that categorized in the moderate killing activity. Okay, and also we determine the effect of the immunotoxin and the binding with the albumin on the cytotoxicity. And we found that after binding the immunotoxin EA version with the albumin, the binding of the two protein not interfere the cytotoxicity of the immunotoxin. Okay, so at this point, we can summarize that the EA linker still have the binding ability to albumin and no effect on the cytotoxicity activity. To conclusions, the EA with the rigid linker exhibits the lower expression yield when compared with the GS format. But the EA linker format show the binding ability to the folate alpha receptor and human serum albumin higher than the GS version. At this point, we can conclude that the type or the type of the linker affect the expression yield and affect the binding ability to its antigen. Okay, and to in vitro investigation of the cytotoxicity, we show that our immunotoxin revealed the IC50 value in the nanomolar range for the variant folate alpha receptor positive cell line. Okay, however, our immunotoxin exhibits an approximately 200-fold lower cytotoxicity when compared with the previous published immunotoxin based on the same toxin PE24X7, which may attribute to its ability, which may attribute by the ability affinity that difference, internalized rate that different in among the antibody as a targeting molecule and the surface antigen density that can affect the cytotoxicity of each immunotoxin. To translate this immunotoxin to the further clinical use, the first one we need to improve the antibody affinity within the moderate affinity into the higher affinity to mediate the higher rates of the internalization. And the next one, we need to prove or determine the in vivo half-life test after it's bind with the serum albumin in the blood circulation. And the last one, we need to evaluate the tumor killing activity of the immunotoxin in mice or in vivo. Okay, that is my project that has been done so far. And the last but not least, I would like to thank the funding from Asahi Glass Foundation again, and thanks my lovely colleague to make this project possible. Okay, that is my project that have done so far. Are there any questions from the floor?
Any question ครับ May I ask you one question ครับอาจารย์กนิกา regarding to the study of comparison between use like a standard specific like a monoclonal antibody folate alpha receptor or another one that may be close to clinical use like the ADC technique and this immunotoxin technique. What is the add-on benefit of this immunotoxin comparing to the ADC feature that may already some clinical proof the benefit in terms of the cancer treatment? Yeah. What is the
Okay.
Yeah.
Okay, when compared with other types of antibody drug conjugation, I think it depends on the type of cancer and the type of drug or toxin that we use. Some cancer, for example, in the cervical cancer or lung cancer, the toxin, the PE toxin, very high potent to kill the cancer. So if we develop the immunotoxin to increase the specific of toxin to the cancer, can increase the therapeutic effect to the patient. I think it depends on the drug and the toxin that you have. If colorectal cancer, some drug has the potent for the cure cancer. It not if the toxin have potent to kill the cancer. So we go with the other type of the antibody drug conjugate. For example, load that drug in the immunotoxin and conjugate load that duct in the vehicles for asymptotic liposome and tag with the antibody to create ADC duct conjugate. I think it's best for colorectal more than the use the immunotoxin. I think it depends on type of cancer and depend on the toxin that can affect on that cancer.
Thank you, because I think in lung cancer, as you may know, that we have many targeted therapy that quite effective, but we also have some patients that unfortunately do not respond to the targeted therapy and the disease progression. So I think this might be the target group that you maybe in the future, if you can develop the experiment to test in this kind of setting, and also maybe close contact with some the pulmonologists or oncologists who work in this field and may shape your drug targets to be more specific in this population, because this is quite relevant to the clinical setting to use this immunotoxin.
I hope so. And this, my antibody now, my colleague used this VH antibody as the antibody on the CAR T cell, and they successful to kill the xenograft mice of the ovarian cancer. So it's have our VH have the potential to in many aspects of the clinical use.
Thank you, Kap. So
okay, thank you.
To the end of the session, and thank you all the presenter to provide a very, very wonderful session. And I would like to close the session. Thank you very much.